and enter your queries or any issues regarding audio and video in the chat box. So your concerns will be addressed. So now I am requesting Kamala Golla ma'am to take the session forward.
Thank
Thank
you,
you all.
Bhavani. Thank you. Yes, good evening all. Am I audible, students?
You are audible, Kamala ma'am. You can continue.
Yes, ma'am. Just I want to share my screen. Is
Your slides are visible, ma'am. You can put in slide mode.
yes, ma'am. Yes. 1 second. Good evening all. Shall I move on to today's session is protein structure prediction, tools and databases. So here today topic is what are the different tools utilized for to predict the structures as well as what are the databases available regarding protein related informations, right? Right. So why we should know about protein structures? What is the importance? So many databases are there, so many prediction tools are available. But by knowing of the structure, what is the benefit we are getting? So these are the uses by knowing of the protein structures. Okay, 1 second. So first one is to understand the protein function. Structure, by knowing of the structure, what kind of amino acids are there, what is the exact structure. By knowing of this, it is what kind of functions are carried forward by this protein. We can analyze or we can understand the things. For example, in a cell, so many proteins are present in cytosol. Some of the proteins are present in mitochondria. Some of the proteins are present in nucleus and nucleolus, right? So, if at all the proteins which are present in the cytoplasm, most commonly they take a part in the metabolism like glycolysis, etc. In case of mitochondria, they carry forward the TCA cycle and electron transport chain. Okay, these are the well-known metabolisms. That is the reason I just mentioned those things. So, to understand the functionality, so catalytic function or carrier function, if the protein is present in the membrane, that is facilitating transport of respective molecules either into cytoplasm to into the cell or outside of the cell, right? So if the protein is not permeable to the membrane, it is passing the signal. So this kind of, see, by knowing of the structure, what kind of function is carried forward in the cell, that should be knowing. Okay, that is the first thing. Second one, drug discovery and design. So for example, if any cancerous cells are there, that means maximum proliferation is in place, right? So any drug about cancer treatment and all, so that drug should be targeted the respective protein which is not attaining the cell normal phase to proliferative phase. So these kind of drug discovery, not only cancer, any kind of drug disease caused by any external element or internal elements. That means pathogen causing diseases. For example, any fever, cough, cold, anything, right? But it is caused by an infective organism, right? So the drug targeting system or drug designing system means here we are designing a drug which destroy the pathogen or which destroy the. Yes, please mute yourself. Right. So now the thing is drug discovery means respective organ which is causing disease inside of the body, that is whether we are destroying the pathogen or the entry of the pathogen is destroyed. So in this way, by knowing of the path, by knowing of the structure, we can design different kinds of drugs. Okay. And studying diseases. Okay. So many diseases caused by so many number of proteins. Okay. For example, prions. Do you know this word, ma? Prions.
Yes, ma'am.
Yes. What is the importance of this? Prions means anyone. Give me one word. Prions. Anyone? Prions. Anybody know this word? So, for example, mad cow disease is caused, right? It is one of the structural alteration of a protein, small protein molecule, right? So, studying of diseases. So, by knowing of structures, we can study the different kinds of diseases, what exactly happen or what exactly modifying in your body. Engineering proteins, it is the one most beauty of this engineering proteins. Engineering proteins means, for example, I'll always go with the insulin, right? So insulin usually produced in human body, right? So for treatment of diabetes, we are producing insulin. Are you extracting insulin from the pancreas of the human body or any mammal? Yes, ma'am. Please tell me the answer. Are you extracting insulin from the human pancreas or any mammal pancreas? No one else, please put any word in the chat box. Yes or no? So initial stages, we are extracting insulin from pork liver. Sorry, pork pancreas. Okay. Later on, this is modified. So rDNA technology, protein engineering. Okay. These are in place. So now whatever the protein we required, okay, you just construct a gene and introduced into the respective host, whether it is prokaryote or eukaryote. If it is prokaryote, that is E. coli, and eukaryotic expression system, it is maybe Saccharomyces cerevisiae or Pichia pasteuris. So many strains are in use, right? So we just keep that gene into those organisms and we express the gene with the desired protein, what exactly you want. Okay, so nowadays we are using insulin production through protein engineering strategies, right? So to engineer a protein with the help of desired sequence or by knowing of the structure. Remember guys, first we know the structure, then we can plan and execute the expression of the particular protein either in prokaryotic system or eukaryotic system. This is the importance with respect to knowing of protein structure. Designing of biomaterials, right? So biomaterials means again these are part of the drug delivery system, right? So drugs are capsulated with the biodegradable materials, right? So by knowing of the structure, we have to inactivated proteins used as a coats for the drug. Okay, biomaterials for filtration purpose of waters and whatever the things. So we have to use different materials as to filter the contents, right? So in that by knowing of the protein structure, we can easily use to construct the biomaterials. Okay, next, to understand the molecular mechanisms, right? By knowing of the structure, what kind of reactions are taken by that protein. For example, electron transport chain, okay, with the help of complex number 1, 2, 3, 4. Okay, oxidative phosphorylation is carried forward by complex number 5. But glycolysis, glucose conversion to glucose 6-phosphate with the help of hexokinase enzyme, right? This hexokinase enzyme is present in cytosol. This ATP synthase is present in inner mitochondrial membrane, right? Understand by knowing of the structure, we can easily understand the molecular mechanisms of that particular protein. Is it clear, ma? Is it clear?
Yes, ma'am.
Right. Personalized medicine. Right. So, for example, for overall group of people are treated with one kind of drug, but the response is different from person to person. In that case, personalized treatment with the desired medicine. That person is respond to which kind of medicine. We can predict by knowing of the proteins, which proteins are highly increased in serum. So proportionally, we can design certain medicinal use to the respective individual. Personalized medicine, it is also generated by knowing of the protein parameters or the important parameters which are elevated in the blood. For example, liver abnormality is there in the individual, SGOT, SGPT, gamma glutamyl enzyme. Okay, these kind of enzymes are increased in the bloodstream, right? So that person is suffering with liver related abnormality. Next, the person creatinine levels are elevated, right? The person is suffering with kidney related issues. Okay, right. So these kind of disease identifications happened by detecting the respective enzymes in the bloodstream, right? So then when you have to go with the treatment, we just personalize the treatment. That means liver abnormality person is treated with respective medicine which is treat the liver illness. Okay, that person is treated with the different kind of combination of components, right? So kidney abnormality person is treated with different kinds of medicines, right? So by knowing of specific parameters in the bloodstream, it is also helpful for treat the respective diseases in the respective individuals, right? And one most important point, studying enzyme catalysis, right? Basically, all enzymes are proteins. All enzymes are proteins, but not all proteins are enzymes, right? So enzyme means most of them, right? I'm not saying 100%. Most of the enzymes are made up of proteins. By knowing of the structure, what kind of reaction is catalyzed by the enzyme is known by the individual. Is it clear? Study of enzyme catalysis. Enzyme is nothing but biocatalyst. It can be able to catalyze a reaction. Substrate component is converted into product. By knowing of the enzyme structure, which kind of reaction is carried forward by that enzyme, we can easily notice. That is one more importance, right? And improving agriculture practices. Remember, guys, pesticides. We are using n number of pesticides to kill the pathogens in the respective agriculture field, right? So to kill those pathogens with the targeting of respective proteins, right? And one more thing, improving agriculture practices, that means yield. Once you know the protein which is responsible for the production or yield improvement, okay, we just use the triggering points and enhancing those protein production in the respective crop. Okay, automatically we improve the agriculture production. Is it clear, ma? Is it clear? Yes, ma. Right. Now we just move on to the protein structure prediction methods and softwares, right? So before entering into this, protein structure levels are expressed in 4 ways. One is first one. Proteins means what is meant by proteins, ma? What is meant by proteins? What is meant by proteins?
Amino acids.
Okay, group of amino acids are connected with peptide bond, right? So here levels are explained with protein structure, primary structure, secondary structure, tertiary structure, and quaternary structure. First one, primary structure. So in this, here what exactly explained?
Kamala ma'am,
Yes, ma'am. Yes, ma'am.
suddenly it stopped.
Okay. I just want to show one image. I want to
Okay,
try. Yes.
okay,
One
no
second.
issue.
Yes, ma'am. One second, ma'am. One second, ma'am. Give me 1 second. I'll show it. Okay. Are you able to see the screen, ma'am? Are you able to see the screen? Yes.
Yes, Kamala ma'am, you can continue.
Right. So now the thing is primary structure explain the sequence of amino acids, right? So here what exactly happened? Right? Amino acids are connected with peptide bonds. Amino acids are connected with peptide bonds, right? So usually amino acids contains amino group and carboxyl group, right? So the first amino acid connected with the next proceeding carboxyl group of the next amino acid, or otherwise first carboxyl group of the amino acid connected with the next amino group of the next amino acid, right? So in between carboxyl group and amino group, this peptide bond is happened, right? So amino acids are connected with each other through peptide bonds, right? So first amino acid carboxyl group is participating in the peptide bond. The respective terminus is denoted as N terminus, right? So the ending terminus is denoted as C terminus. Usually amino acids are connected with peptide bonds. Okay. So it starts with the N terminus. Obviously it ends with the C terminus. Is it clear, ma? Is it clear? See, ma? The amino acids are arranged through peptide bond. Amino acids are connected with the peptide bond. So see, amino acids are connected with peptide bonds. So one terminus is denoted as N terminus, another terminus is denoted as C terminus. So primary structure explains towards the peptide bond and molecular weight. Right? Molecular weight and how many amino acids are there, their charges, etc. Right? So now I am explaining insulin amino acid sequences as well as disulfide bonds and intradisulfide bonds. I am taking the example of amino acid structure of the insulin only. This insulin having A chain and B chain, interdisulfide bonds and intradisulfide bonds are there in this structure. So now we just move on to the next. So how to predict the primary structure of any sequence, right? So in website, students, please use this one. Potparam, P-O-R-T-P-A-R-A-M, param, right? So please use this tool. It is available in online, right? you can paste your sequence. You can paste your sequence. Okay, only amino acid sequence. Remember, guys, FASTA sequence is not accepted by this web page. Is it clear, ma? Primary structure predicting online tool. Okay. Port param. Is it clear? Are you able to? Okay, I will show it to you. 1 second. Okay, so port param, right? This is the home page. Are you able to see the screen, ma? 1 second. Stop sharing. Yes, ma. Are you able to see the screen? Right, students?
Yes, ma'am.
Yes, right. So port param, right? So now I am taking a sequence. 1 second. I'll show it to you. Okay. Right? This is the FASTA sequence. I'm taking this one. I just to go with the okay. I want to paste it, right? So now I'm pasting the FASTA format. Please notice this thing, right? Compute the parameters. Sorry, your sequence should be at least 5 residue long. Please try again, right? I'll just go to back and I am removing the FASTA information. See? Okay. Just I am pasting the amino acids, right? So this tool helpful to compute the various physical and chemical parameters for a given protein. Remember, guys, this tool helpful for to carry out the various physical and chemical parameters for a given protein. But this protein sequence must be in amino acids only. Don't give the FASTA sequence, right? Okay. So now I am pasting one amino acid sequence and click the compute the parameters, right? So total amino acids, number of amino acids. Are you able to see the screen? Yes, ma. Are you able to see the screen?
Yes, ma'am.
Yes, sir. Yes. So now see, I just provide one amino acid sequence. I don't know the number of amino acids, right? So this is giving total number of amino acids present in the query sequence, that is 28. Molecular weight 3139. Okay. Theoretical pI, isoelectric pH of the sequence 6. Is it clear? What are the amino acids are present and what is the composition and total number of negative charge residues, total number of positive charge. See, total number of negative charge residues, aspartic acid and glutamic acid, 3 are available. Right, total number of positively charged residues, arginine and lysine, 2. Okay, atomic composition, formula, total number of atoms. Okay, extinction coefficient, everything, physical and chemical parameters. Okay, estimated half-life of the sequence, instability index, aliphatic index, and hydropathicity. Okay, these are the important physical and chemical parameters. We can utilize this for further construction of a protein or further involvement of drug protein interactions and all, etc. Right? So the primary structure is consisting of amino acids with N terminus and C terminus, right? So while you are using that sequence and we can get the physical and chemical parameters using this tool. Is it clear? Are you able to? Yes, ma'am?
Yes, ma'am.
Right. So please do one exercise. Just paste according to your wish some alphabets. Okay. And try to get the output from this tool. Yes, quick. Do it fast. Do it fast and post it. Take the screenshot and post it. Any amino acid sequence, no issue. Do it fast. So this is the information regarding primary structure. So by using our primary structure, we can get the physical and chemical parameters such as how many number of amino acids present in the sequence, molecular weight, and atoms. Okay. Next, negatively charged amino acids, positively charged amino acids, extinction coefficient, estimated half-life, and instability index, as well as hydropathicity. Is it clear, ma? Are you able to follow this one? Please use this tool and get the information quickly, fast. Are you able to follow this one, ma? Yes. Anyone completed? Please show it to me. Anyone
Somebody
completed?
has sent the image. Where did you send the image? Is it in the chat box?
Yes, yes,
They
yes,
send the
yes.
message in chat box. You can have a look at it. Kamala ma'am.
Yes, ma'am. Yes, ma'am. I'm seeing. Yes, yes, yes. Okay, good, good, good. Okay. Yes, ma'am. Thank you. Right. Shall I move on to next, ma'am? I'm very happy. I'm getting so much of results and all. Yes, thank you, thank you, thank you. Right. Okay. Now I just move on to the next one. Okay. So in that way, we have to detect the physical and chemical parameters using primary structure. One of the online tool is Param. It is available in the internet. Okay. Right. Next one. Next one. Second. Okay. Now, secondary structures. Secondary structures. Primary structures consisting of amino acids. That's all. Right. What? Negatively charged amino acids, positively charged amino acids, and different kinds of amino acid composition we can know by the primary structure. So, due to presence of these amino acids, proteins get different conformations that are resulted in the secondary structures. Remember, secondary structures usually either alpha helix or beta plated sheets. Okay, alpha helix or beta plated sheets. Okay, so these alpha helix or beta plated sheets are connected with beta bends, and beta turns. These are the 4 important secondary structures we should know in the protein structure. Secondary structures means what kind of amino acids present in the protein that is known in the primary structure, right? Due to presence of that particular amino acid, the protein is either configured with the alpha helix or beta plated sheets, beta bends, and beta turns. These are the different strategies with respect to secondary structure of proteins. Secondary structure of proteins means may have the either alpha helix, beta plated, beta bends, or beta turns. Alpha helix is of 2 versions: right-handed alpha helix, left-handed alpha helix. Beta plated sheets, they are parallel, anti-parallel, right? So here right-handed helix is visualized in your structure. This is alpha helix. Anti-parallel beta sheet is resulted in this. So alpha helix and beta plated sheets are connected with beta bends and beta turns. This is beta bend and this is beta turn. Is it clear, ma'am? Secondary structure majorly consisting of alpha helix and beta plated sheets. These are connected with either alpha bends or beta bends or beta turns, right? So this is a brief about secondary structure of protein. Now, how to predict the secondary structures using online tools, right? Next, SEMA, this is CFSSP, Chow-Passman Secondary Structure Prediction Server. Okay, this server predicts the secondary structure of protein from an amino acid sequence. So here the important thing is we should submit FASTA formatted sequence only. Is it clear, ma'am? So it is a secondary structure predicting server present in online. This is CFSSP, Chow-Passman Secondary Structure Prediction Server, right? So by using of this server, we can able to predict the secondary structure. Okay, it is following Chow-Passman algorithm. Is it clear, ma'am? Are you able to follow this one? So I just want to move to the Windows. Okay, right. Now I just move on to the right. So you just biogen.org/tool and CHOU/Passman bar index. I just post this link, this web link in the chat box. Give me 1 minute. Okay, please use this link. Okay, right. Now I just want to paste the sequence. Okay, predict. I just click the predict, right? So now my target sequence consisting of 70 amino acids, right? So it consisting of helix, sheet, turn, and coils, right? Secondary structure consisting of helix, sheet, turns. So combining of this, it is consisting of, right, helices, sheets, turns. These are the secondary structure of respective polypeptide chain or a protein. Okay, whatever query we are putting in this, it should be in FASTA format. Okay, you just keep your FASTA format sequence in the respective paths and run the program, right? Immediately it is displaying the secondary structures which are comes under helix, sheet and turn and coils. Is it clear, ma? Are you able to follow this one?
Yes, ma'am.
Right. So it is also giving total number of residues, how much percentage of helices is present, how much is in sheet, and how much turns are existing in this. The total percentage is expressed in down of the secondary structure details. Is it clear? Yes, students? So please try with your any example structure or FASTA sequence. Keep in the server and get the output. Please post it in the chat box. Do it fast. These are online servers only, so it is easy. Yes, ma, do it fast. Take any FASTA format or FASTA sequence from your database and paste in this server. Okay. Show me the output or paste the output in the chat box. Do it fast. Thank you, sir. Students, quickly post the information. Fast. Thank you so much. Right. So now we can get the primary structure information and secondary structure information by using these online tools, right? Shall I move on to next?
Yes, ma'am.
Right. Thank you. Right. So till now we just use primary structures and secondary structure predictions, right?
Yes, ma'am.
Right. Okay. Now just move on to the next. Next one is tertiary structure and quaternary structure. So there is no much difference between tertiary and quaternary structures. Why I'll explain, right? So in the primary structure, we should know about the sequence of amino acid, right? In the secondary structure, hydrogen bonds, how they are tightly associated with the structure and able to resulted either alpha helix or beta plates, right? Due to interaction of hydrogen bonds, the protein or the primary structure is folded to either alpha helix or beta plated, right? Okay, not only peptide bonds, hydrogen bonds plays a major role to get the secondary structure of a protein, right? After that, tertiary structure, that means compressing of this entire structure with a domain, three-dimensional folding pattern. Remember guys, tertiary structure is nothing but three-dimensional folding pattern of the protein. Three-dimensional folding. What is the importance of this? Right. Here, different non-covalent interactions. Remember, first primary structure is resulted with the help of amino acids participating in the peptide bond, right? They are involved in amino acids are participating in peptide bond formation. That is a covalent interaction. Okay, next secondary structures and tertiary structures are maintained with the help of non-covalent interactions. Those are hydrogen bonds, ionic interactions, hydrophobic interactions, van der Waals interactions. Okay, these are all our non-covalent interactions, ionic interactions, hydrophobic interactions, okay, hydrogen bonds and disulfide linkages. These are all our non-covalent interactions. With the help of these non-covalent interactions, proteins get a three-dimensional folding. That means, for example, 1000 amino acids containing protein occupying 1 meter of area, right? 1 meter area is occupied by 1000 amino acid containing polypeptide chain while you are placing in a linear arrangement, right? It is normally in the primary structure, it is occupying that much 1 meter space area, right? So after it undergoes three-dimensional structure, it is not using 5 to 6 centimeters of area. For understanding purpose only, I am saying this, right? So three-dimensional folding means area occupied by the protein is completely decreased by this three-dimensional folding of a protein, and it is giving a close contact. Hydrophobic amino acids are placed the interior of the protein, and hydrophilic amino acids are placed on the outer surface of the protein. In that way, a protein existing in the cytoplasm. Why means cytoplasm consisting of 60 to 70% of water. So the protein having hydrophobic amino acids and hydrophilic amino acids. Okay, so the surface place is occupied by hydrophilic amino acids. Interior space is occupied by hydrophobic amino acids. So in that way, the protein is present in the aqueous environment or cytoplasmic environment and carry forward the remaining metabolic activities by that particular protein. Is it clear, ma? Tertiary structure. Why? What is the importance of this? Yes, ma'am.
Slide is not being shared.
Showing this sharing. Okay, one second. Yes, ma'am. Now?
Yeah, now it is visible. Please carry on.
Right? So this non-covalent interactions and all resulting three-dimensional folding pattern of the protein. So this three-dimensional folding may helpful for the retaining of the structural functions of that particular protein in any kind of environment. For example, aqueous environment containing all hydrophilic amino acids, right? But usually proteins consisting of hydrophobic and hydrophilic amino acids. Those hydrophobic amino acids, by these foldings, just projected inside of the protein, and hydrophilic amino acids projected outside of the protein, right? In that way, a protein is existing in the cytoplasm of a cell. It is one way of approach with respect to hydrophilic environment. For example, the protein is present in the membrane. So hydrophobic environment is exposed to the lipid bilayer and hydrophilic expressed into the interior space of the protein, right? So in that way, according to the functionality, this domain plays three-dimensional folding plays a major role to carry out that respective functionalities of the protein. So this three-dimensional folding is denoted as domain. Domains are commonly seen in the structures, tertiary structures, right? So domains are most commonly stabilized by non-covalent interactions, right? So single domain is denoted as tertiary protein structure. Multiple domains associated with the protein cluster, that is nothing but quaternary structure. For example, tertiary protein, example myoglobin, it is having single domain. Okay, quaternary structure protein, example hemoglobin, it is a tetramer.
Kamala? Kamala? Kamala?
Madam? Madam, am I audible?
Now you are in audible.
Yes. So this structures. So first we go with the primary structures and secondary structures. Then primary structures, secondary structures, respective database tools, online tools, which we can predict the primary structure information and molecular weights and everything done by different servers which are available in the internet, right? Now we just move on to the tertiary structures and quaternary structures. Okay, so tertiary structures and quaternary structures are the functionable protein structures in a cell, right? Now we just move on to the next. So different databases which are available in the internet. First one is protein database. Protein database, right? We already know with this protein database, PDB database, right? So protein data bank, right? So we can search any kind of protein by using this. Already I explained in the previous class. So we just go with the according to your interest. Then you have to type any name. Okay, you can get the similar structures in this database, right? I just already explained this one. So we can download the different formats, right? FASTA sequences, PDB formats, everything. So this is one of the database with proteins. Okay, protein related information is present in this, right? Next database. Okay. This is SCOP database, structural classification of proteins. This is another database. Okay, structural classification of proteins information is available in this database. Okay, I'll show it to you. Right? So, see, SCOP, you just type SCOP and database, then you can get this structural classification of database, right? So this database usually we can use for similar, structural similar proteins. Okay, we just check the structural similar proteins by using this database. And also it is able to share common evolutionary origins, right? So this database, okay, which is giving an information regarding, see, structural classes. We can use this browser for structural classes. So alpha proteins, beta proteins, alpha beta proteins, alpha beta proteins. See, alpha beta proteins in both are different, right? So proportionality, which is competing with small proteins, everything regarding structural concepts. Okay, we can search with the structures. We can go with the protein types, right? With the structures, secondary structures and alpha, beta is the most important. So those, based on that, we can search the proteins here. And another thing, due to presence of alpha beta chains, the protein giving a respective structure, that is globular proteins, which are present in the cytoplasm or any liquid portion, either mitochondrial cytoplasm or normal eukaryotic cytoplasm or interior surface of endoplasmic reticulum, and all this globular protein information we can browse by this tool, right? This server. Membrane proteins, fibrous proteins, non-globular proteins. So different kinds of proteins we can search by their structure and by their secondary structure and final structures, right? So whether they are associated with the cytoplasm or they are associated with the membranes or they are in a fibrous in nature. Okay, fibrous proteins, muscle related proteins or cytoskeletal proteins, all are the fibrous proteins, right? We can browse that kind of proteins by using this server. Not only PDB, different servers are available. This is another server, structural classification of proteins, right? So it is giving a clear-cut structural similar protein search by using this database. Okay? Yes, ma'am. This database is providing a comprehensive description with a structural and evolutionary relationship between all proteins with the search one, right? So if you are searching any protein in this, okay, it is giving a structural information as well as evolutionary relationship between the resulted proteins. Okay? Yes, ma'am.
Yes, ma'am.
Right. Okay. So here, what is the important thing, right? Keyword. Okay. So for example, I'm using insulin, right? See? Insulin, I'm placing, right? Insulin family, right? Functions. Okay. So protein, insulin, Homo sapien, right? So ID, this is the ID provided by this. It is a link with Uniport, PDP, RCSB, PDB. Is it clear, ma'am? Is it clear?
Ma'am.
Right. Seema? Okay, the amino acid sequences. Just we have to insulin. We just highlighted insulin, right? The respective continuity of the amino acids, right? This domain details, everything, okay, is displayed by this search engine. Okay, other domains for this sequence. See, external links, super families. See, phylogenetic relationship with the different similar proteins. So this web server give the detailed information about protein and similar proteins and phylogenetically they how related to others, right? So this is a database is completely providing information regarding structural and evolutionary relationship. Is it clear? Is it clear, ma'am?
Yes, ma'am.
So you can go with the, see, either structural class. If you are clear about the protein, that is an alpha protein, right? So we can search with the all alpha proteins, or your protein is a globular protein, right? You can go with the globular proteins, right? If you know the structure, you go with the respective log or respective extension. Is it clear, ma'am? Yes?
Yes, ma'am.
Right. So another database, SCOP, Structural Classification of Proteins. Okay, structural similar proteins are analyzed by using this database. Is it clear? We can find out the structural similar proteins, and their evolutionary relationship is also explained by using this database. Is it clear? Shall I move on to next?
Yes, ma'am.
Right. Okay. Right. Are you able to see the screen? Yes, ma'am?
We are able Yes, to ma'am. see.
Yes. This is a third database, CATH database. Class, architecture, topology, and homologous superfamily details. Okay. This database predicted structural domains, okay, which are available in this, okay, and it is also providing class, architecture, topology, and homologous superfamilies. Is it clear? It is holding 151 million protein domains. It is holding 151 million protein domains, okay, under different superfamilies, right? Yes, ma'am. CATH database, class, architecture, topology, homologous superfamily. Is it clear, ma? Is it clear? Shall I move on to next? I just want to share the, one second. I want to share the Google, one second, right? Okay. Give me 1 minute time. Okay, protein structure classification database. CATH means protein structure classification database. It is a comprehensive resource for protein structure classification, right? Okay, it is providing evolutionary relationships. It is very similar to scope, right? But what is the difference? Later I will explain, right? It is also explain the evolutionary relationships, 3D structure, and functional similarities. Remember, previously scope is giving the phylogenetic relationship and domains. Okay, similar domains. But here it is giving functional similarities, right? I just want to click this one, right? Okay. Okay, so see, once we click this one, we are getting 3D structure. Find out the 3D structure of your protein adopted. Is it clear, ma'am? Yes? 3D structure prediction is possible by using this server. Protein phylogenetic relationship with the different similar proteins is feasible with this. And functionality of the protein, conserved sites. Okay, protein function is also predicted by this, and conserved sites we also predicted by using this. First, 3D structure, protein evolution. That means how it is correlated with the different proteins, how it is phylogenetically related with the remaining proteins, protein functions, and conserved sites. Okay, is it clear, ma? Is it clear?
Yes, ma'am.
But here one thing I will tell you, such word is not that much easy. We go with the IDs. Okay, Uniport IDs or PDB IDs. Any ID is important to search this engine. Is it clear, ma? Is it clear? So database search is feasible by using 3 different databases. We can search the proteins using 3 different databases. One is PDB database, scope database, AS database. Is it clear? 3 different databases used for search to get the suitable proteins, right? If you search any protein in this, so first PDB database, 3D, 3-dimensional structure, and what kind of PDBs and all, everything information is there in PDB database. Scope, that means phylogenetic relationship with respect to that protein is also given in that server. Okay, next one, catch. It is a class, architecture, topology, and homologous. The words itself give the information. Which class of protein? How it is evolutionary interlinked with this? What kind of functions? Okay, what kind of architecture is there in that protein? And how it is belongs to which kind of superfamilies? Is it clear? These are the 3 different databases. Are you able to follow this one?
Yes, ma'am.
Right. Okay. Second, give me 1 second. Till now, what kind of information we are getting by this workshop? First thing, primary structure prediction, secondary structure prediction. Third one, 3 different databases. Okay, we can search the protein by using 3 different databases. Is it clear, ma? Shall I move on to next level?
Yes, ma'am.
Yes, right. Thank you, guys. Okay, right. Are you able to see the screen?
Yes, ma'am.
Right? Yes. So now we just move on to the predict to create a structure, right? Homology modeling. That means you are giving a query sequence to the computer, okay? Using database, that computer able to generate a valid structure of your query sequence. This is simply denoted as homology modeling. Right, ma? Homology modeling means you know the amino acid sequence. Okay. So you just put that amino acid sequence in the FASTA format. Okay. Just keep this information in any online tools. That means you are submitted to computer, right? This undergoes homology modeling using different databases or available information and executed as valid structure of that respective sequence. This is simply denoted as homology modeling. Is it clear, ma? Is it clear? Yes. Yes, students, are you able to hear my voice? One second.
Yes, ma'am.
Yes, right. So homology modeling is nothing but it is not a big task, ma'am. So first you should know about the FASTA sequence. Ma'am, I don't know that about FASTA sequence and all. You know the amino acid sequence. Okay, and put a symbol. Then you keep that sequence into any online tools available in computer. Then it is generating a three-dimensional structure of the respective sequence. How this is going to be happened? It just utilize the existing databases and existing similar structures and just pull up the structures into one online version. Then you just give the output of valid three-dimensional structure of the respective query sequence. Okay, so now just move on to the next. Right? So Swiss model, predicting the protein structure using Swiss model, first one, right? So how to use this Swiss model? Just to go to the internet, web browser, right? Just type the Swiss model, all capitals, SWISS model, right? And then next, second step, just put a FASTA sequence of amino acids, right? FASTA sequence of a protein. You just give this as an input. Yes, ma'am. FASTA sequence of a amino acid or a protein. Remember, FASTA sequence of the protein. Just give an input to that browser, right? And then you just select the modeling options. Here, two modeling options are there. One is automated modeling and the second one is template search and selection. If you know the templates, go for the next level, template search and selection. Or otherwise, we just trial and error basis, you go with the automated modeling. Okay, then press next. Okay, so it takes little bit of time and execute the three-dimensional structure of the protein. I'll show it to you. 1 second. Right. One second. Okay, so this is the thing, what exactly we just type in the web server, right? So now here this box is explaining paste your target sequences, right? So I'm taking this sequence. And paste here. Okay. Okay. So the target sequence must be longer than 30 residues, right? So I don't have 30 residues. That is the reason I just want to paste the same sequence and remove this one. Right? Okay. Upload the target file, right? So see, search for templates. I already explained this. Search for the templates or otherwise build model, right? Okay. So I just want to remove this one. Okay. So automatically it is, if the sequence is more than 30, it is going to the next level, right? So now we want to search for the templates or we go with the build model, right? So first I want to search for templates. Templates means which are very similar to the sequence, right? See, we are searching for templates. It will take little bit of time, right? Build the model. See, template results are still running and simultaneously another block we are generating the 3D structure of the respective query sequence. So basing on the amino acids, okay, so more number of amino acids, more number of execution of 3D structure or templates for the respective query sequence. Remember guys, less number or more than 30 amino acids are mandatory requirement to generate 3D structure of a protein by using Swiss model workspace, right? So, but here more than 30 is mandatory, right? So if it is 1000 or above, right? So it is not able to predict the 3D structures appropriately, right? So better you have to take minimum amount of amino acids, minimum number of amino acids. We get good resolution with respect to that three-dimensional structure of the protein, right? So now my model is generated, right? So here for further works, we can download the structure. Here download files are there, right? Okay, either you have to PDB format, okay, deep view format, whatever you require, you can download that and you store this file. Okay, so here two mini models are generated. See, this is the model 2, this is the model 1, right? Okay, magnification, minimization, everything is feasible when this, right? So two models are generated till now. Okay, up to 5 models also generated, right? So in this Q mean Z score, insulin like growth factor. So we just take this one and paste in this. Okay, we can get the 3D structures according to your comfortableness and convenience. We just do the next level of work by downloading the files. So usually we have to download in PDB format. Okay, so now we just go with the templates. How many templates are there? So see, here these many templates are available, right? So green color is similar sequence, right? So the white color is differences with the query sequence and existing sequence. Remember, right? Similar sequences, similarities. See how much similarity structure is there that is expressed with the respective query sequences, query structures. Okay, these many query structures are resulted. Okay, sequential identity. Okay, sequential identity. We can get, okay, next topology. Interface similarity. Okay, so these are the different things with respect to the protein. If we go with the templates, okay, maximum number of templates are resulted in this. So which one is showing maximum similarity? We just used as that to develop the further structure, three-dimensional structure of the protein. Is it clear? Are you able to follow this one? Are you able to follow this one, ma? Swiss model. Please try with any kind of sequence and execute the models fast. Do it fast. Any query. For PDB database, you just retrieve the FASTA sequence, which must be having more than 30 amino acids, and paste in Swiss model workspace and just generate the 3D structure of the respective sequence. Please do it fast.
Should
Srinidhi, good. Anyone completed? Please post in chat box. Okay, sir. Getting slow. So this is guanine nucleotide binding protein. Okay, good. Thank you, Mahesh. Everyone is working with this. Nice. 2 results. Okay. Very good. Yes. Yes. Everyone able to follow this one? Please give a response in chat box. I just move on to next. Okay. Next. I just want to move on to the next. Hmm. Right? So we just go with the Swiss model, prediction of protein structure using Swiss model. Okay. So we have to download the files as per 3DB PDB formats and used for further analysis. Okay. Next one is prediction of protein structure using predictprotein.org. Predictprotein.org. Right? You can type this predictprotein.org in web browser. Okay. So you just paste the amino acid sequence, choose the predict option. But here, select the desired type of prediction, whether you are requiring, whether you want secondary structures or 3D structures, functional site notion, disordered prediction, right? This is a different browser. Okay. So protein structure prediction. Okay. Now I just want to. Okay. So this after selecting of your desired thing, then you have to submit your query sequence. Okay. This tool is analyzing your input and display the results. But what kind of parameters you are looking for? So this is proportional to the time course, right? Time varies depending on the length of the amino acids and selection of the options. Remember, previously in Swiss model, we just predict the template queries, right? It will take little bit of time. And development, model development, it is just with a little bit of time course, it is giving the 3D structure of the protein, right? Same version, what kind of sequence we are using as a query sequence. And second one, selected options, what kind of options you are choosing. Basing on this, okay, the processing time is vary from protein to protein, right? So once processing is completed, it is display the results. Okay, so we just download the files and used these files for further analysis. Okay, structural predictions and functional data is also valid for further analysis of the particular protein. Is it clear? Is it clear?
Yes, ma'am.
Right. Okay, now I just want to go to back. 1 second. Stop sharing. Then I want to share the windows. Right. Google. Right. Okay. Next, right. Okay. So predict protein structure. Web page is like this, right? So otherwise I will go with the 1 second. Protein. Okay, so predict protein. This is the link we can use. I just want to post in this chat box. Predict protein, right? You just use this link and enter the amino acid sequence. Remember, here FASTA sequence is not required. So you just paste the amino acid sequence, right? No problem. Structure. Display the FASTA sequence. So now I just want to okay, I just want to take the sequence. Okay, copy the paste sequence, and I just want to paste the sequence here. Okay, right? Okay, predict protein. Okay, So here dashboard. Okay. Okay, so in this dashboard, just what kind of parameters we can just one second. Okay, secondary structure. Okay, next protein disorderness, flexibility, functional function annotation. Okay, effect of point mutations. Everything, what kind of information is explained. Okay, what kind of parameters we can analyze. These are all are display on the left side. Okay, so now our Seema, are you getting the results? Yes. Are you able to see the screen?
Yes, ma'am.
Right. Now I just want to paste one sequence. Right. So click here to visualize your input. Click here to visualize a table of aligned sequence with uniport and PDB identifiers. Right. Alignment of your sequence. Visualize the alignment of your sequence with PDBs, PDB IDs. Okay. About the sequence, this kind of further links are displayed in the workplace. Right. So next one, secondary structure. Okay, secondary structure, either helix or plated. That kind of information is displayed in this, right? The entire sequence is categorized into beta plates and every information, right? Relative B values, protein binding probability, DNA binding probability, which domain having DNA binding probability, RNA binding probability, how many disulfide bonds there are there, how many conserved regions. Every information is given in detail with the respective predict protein structure. Is it clear, ma'am? Predictprotein.org. This is an online server which provide different kinds of secondary informations, secondary structure information, and topology related information, disorderness, and protein binding probability. This protein is able to bind to the DNA or RNA. How many disulfide bonds are there? How many conserved regions are there? This kind of information. And also it is summarize the amino acid sequence. Okay. Is it clear, ma'am? Are you able to follow
Yes,
this one?
ma'am.
So it is not giving 3D structure straight away. Remember, this is predicting protein structure, right? But it is giving only secondary structure information. It is not giving three-dimensional structures. That is the difference. What is the importance of this? This is giving secondary structure information as well as conserved regions. So better we just opt which kind of protein we are opting for the further. For example, previously one of the person is using work with P53. So how many conserved regions are there? How much secondary structure is able to interact with the drug? That kind of information we can predict by using this online tool. Swiss model is directly providing 3D structure, three-dimensional structure, but this is only providing secondary structure and different parameters. Okay, and it is also providing 3D structure, ma. Seema, number of PDB matches to this structure. So it is giving matches, and furtherly we go with the further steps, we can get the approximate or exact 3D structure is not display by using this, right? Better Swiss model is giving direct 3D structure, and protein predictprotein.org giving secondary structure of the proteins and different parameters. Is it clear? Shall I move on to next?
Yes, ma'am.
Right. Are you able to see the PPT? Yes, ma'am. Are you able to see this PPT?
Yes, ma'am.
So by using this, we can predict the protein structure, especially secondary structure. Is it clearer? Is it clear?
Yes, ma'am.
Right. Okay. So this is another protein structure prediction online server, right? So I just want to post this one. One second. Post the link. Protein structure prediction server in online. Okay, just to go with this. Second, I just want to post. Yes, please use this link to get this protein structure prediction. Okay, so you just place the amino acid sequence and you just try here. We can get the 3D structure. Alpha fold, right? Alpha fold, protein folding, right? The structure is displayed basing on alpha fold. A transformer based AI model generating system, right? So you just paste the amino acid sequence and using this transformer based AI, it is able to generate 3D structure of the protein. Is it clear, ma'am? Is it clear? Is it clear?
Yes, ma'am.
So here minimum 20 amino acids, only amino acids sequence only, paste here and you can get the structure. Next version is next version is ESM fold. ESM fold, right? So here we can use again sequence or folds. So we can get the 3D structure of the respective sequence, right? So this is using a scores predicted local distance difference test. Using this course, it is able to generate three-dimensional structures. Is it clear? Are you able to follow this one? So this evolution is happening with the respective two scores, PAE scores and PLDDT scores. These two scores cumulatively give the three-dimensional structure of the respective amino acid sequence. So this server generating three-dimensional structure of short chain amino acids as well as long chain amino acids, right? Long chains and short chains, which consisting of minimum of 20 amino acids, right? More than hundreds, right? So these two different work environments. One is giving low number of amino acids 3D structure. Another one, more amount of amino acid containing sequences used here as a query sequences to get their 3D structures. Is it clear? Is it clear? Yes, ma'am. Right. Next one. Okay, this is another database, right? Sorry, protein structure database, alpha fold protein structure database, right? It is also helpful for detecting of or generating of protein structures through online, right? So here you have to search the proteins and get the information with this database. Is it clear, ma'am? It is also helpful for development of the structures by using EMBL EBI. Already we know this one, EMBL EBA for multiple sequence alignment, global alignment, blast, two sequences, local alignments, those kind of things. We just go ahead with this EMBL EBI homepage, right? Okay, so this is a database and it is also helpful for structure development of any desired protein. Is it clear? Is it clear, ma'am? Yes? Yes, ma'am. How to use this database for structure development, right? So you just use this database for development of a 3D structure, right? Okay, so we have to consider, just try once more. Here services are provided. By using those services, we can get the 3D structure. Actually, it does not work out here. I already tried. Just better once you try with this, we can get the structure of a protein sequence. So first you have to prepare a FASTA sequence of protein. You just paste the sequence or upload the sequence in the workspace. Now it is not working for me. Okay, then select whether you are just get the query sequences or you have to obtain the structure of the respective sequence, right? So in that way, we have to predict the structure of the protein by using this database. Okay, usually workspace is provided here. Already I just do it, but now I am unable to. Okay, right. Once you try it and you can get the structures of that particular proteins. Right. Next one more online tool is PHYRE, Protein Fold Recognition Server, Structural Bioinformatics. SEMA, it is a protein structure prediction server. Okay, it is usually used for to construct of a structure of small or few amino acids containing polypeptides into number of big proteins. Here there is a possibility to get structures for few amino acids to hundreds of amino acids, right? It is an online server. So we can take this one and we can paste it any sequence here. SEMA. So I just want to paste this link in chat box. Okay, use this link and see here job name 1. I just want to paste the sequence. Sorry. Just get the sequence, I think, for display. Okay, I just take the sequence. I just paste this sequence here. Okay, or we can paste the files or we upload the files. Anything is okay, right? Yeah. So here also we should paste the. Okay, so we should go with the email address. We are not going with this. Okay, so here this is also giving good results. 3D structure of a protein we can get using this online server. But it will take a little bit of time. Yes, your job is in queue, right? So See how many jobs are submitted simultaneously with you. 12 jobs are running, 6 jobs are query. So expected time outcome is 27 minutes. Okay, within 27 minutes, we get the results. Is it clear, ma? This is also online server or online tool. We can use it to predict the 3D structure of the any amino acid sequence. Is it clear? Is it clear, ma? Yes? Are you able to follow this one? Yes, Mahesh. Are you able to follow this one?
Have given one in the optional job description
Where, ma? What
by
option?
searching, ma'am. You have given one, ma'am, in the optional job description.
I just post the link of that server.
Yes, ma'am.
Hmm.
Yes, ma'am. In the job description, you have given
Okay.
one. You entered one, ma'am. Why?
Yes, ma'am. Why means that is for my option only. We just give the name also. Right. For the sake of example only, I just keep one.
Okay.
Okay. So according to your convenience, you give the name also. Right. Okay. Any
Okay,
doubts?
ma'am.
Now I just stop here. Today class, I will continue tomorrow. Okay.
Can we predict two structures in the same time, ma'am?
Yes.
In one website.
More than two. You can
Okay.
type, but time taking process, right? If you are keeping one
Yes, ma'am.
sequence, within 5 minutes you
It
get the
takes more time,
results.
ma'am.
Ah, yes.
Yes, ma'am.
See, the server also doing jobs, right? So it will
Yes, ma'am.
take time. If you are giving multiple sequences, it will take time, but it display the results, no issues. Okay.
Okay.
So time constraint point of view, I am taking only short amino acids length. I am taking very small sequences, right? If you are okay with that, any kind of protein, for example, hemoglobin, that kind of proteins, anything we can keep it and we can get the unknown proteins. See, what is the beauty of these servers? You can get unknown proteins structures. Unknown protein structures also predicted by using these online servers. Okay, any doubts, ma'am? Anyone having any doubts? Vanita, ma'am, can you please post the attendance, today's attendance, ma'am?
Yes, ma'am.
Yes, ma'am. Anyone having any doubts? Are you able to follow today class? Please respond in the chat box. No, ma'am. What happened? Able to understand. No doubt. Okay, right. Thank you so much.
Thank you, Kamala ma'am.
1 second, ma'am. Vanta, ma'am, shall I wind up the class? Anyone having any
Yes,
doubts?
ma'am.
Yes, Bhavani, ma'am. I just want to wind up this class.
Yeah, yes, ma'am. Yeah. Thank you, Dr. Kamala Gulla, ma'am. And thank you all students. See you in the next session.
Please complete your attendance, ma'am. Just complete
Hello.
your attendance. Yes, ma'am. Mahesh.
Gene symbol, ma'am. How to get the
Sorry.
gene symbol, ma'am?
What
In target
genes?
scan, ma'am. In target scan website.
Target.
Enter a human gene symbol, ma'am. It's giving the example HM G2A. How to get the sequence gene symbol, ma'am?
Please share your screen, Mahesh. Then I will understand the things.
Mahesh,
Okay.
target scan is for miRNA, is it not?
Yes, ma'am.
Right?
Yes, my miRNA.
Oh, okay. Show me the screen.
This one, ma'am.
Yeah, you can take this gene symbol, you can go to gene and put your symbol there, or you can put a microRNA name also there. No, that is the gene sequence. This is from GenBank you are taking. Go to the gene. In nucleotide, you select the gene.
Okay, ma'am.
In nucleotide, go to nucleotide and select the gene there. In that all databases, go to all databases and select the gene. There you select the gene. Instead of nucleotide, you select gene. Trypsin spelling is TRY.
Yes, ma'am.
Yeah. So
Sorry.
that is the, but you have to select human one. Okay, Homo sapiens, you take Homo sapiens, type Homo sapiens, and then you can see when humans, so Homo sapiens
Ma'am,
data is.
Here, enter a miRNA name, ma'am. You can enter either anyone, ma'am.
miRNA ఉండదు ఇక్కడ నువ్వు జీన్ ఎంటర్ చేస్తావు ఇక్కడ ఎంటర్ హ్యూమన్ జీన్ సింబల్ అని ఉంది కదా సెకండ్ పాయింట్
Yes, ma'am.
జీన్ సింబల్ ఎంటర్ చేయాలి ఆ బ్రాకెట్స్ లో ఎగ్జాంపుల్ ఇచ్చాడు కదా అట్లాగా జీన్
Yes,
సింబల్ తీసుకుని అక్కడ ఎంటర్ చేయాలి జీన్ సింబల్
Okay, ma'am.
Mama, can I leave now?
Yes, Kamala ma'am.
Okay.
Thank you, ma'am. Bye.
Thank you.
Participants, I think today also we found only 45 participants, totally 57 have registered for the course, but online only 44 or 45 members are coming, as we have been continuously telling that attendance is mandatory to get a certificate. So I am not sure why the other 10 members